The beginning of an action potential in the sarcolemma and the start of a muscle contraction is called excitation-contraction coupling. Hello, my name is Michelle Cupp, and I will be explaining to you the sequence of events that make up excitation-contraction coupling. First, I will explain a single muscle fiber. One muscle fiber only has one neuromuscular junction. The cytoplasm of a synaptic terminal contains vesicles filled with molecules of acetylcholine, or ACH. ACH is a neurotransmitter, which is a chemical released by a neuron to change the permeability or other properties of another cell plasma membrane. The synaptic cleft and the motor end plate contain molecules of the enzyme acetylcholinesterase, or ACHE, which breaks down ACH. The stimulus for ACH is the arrival of an electrical impulse called an action potential at the synaptic terminal. An action potential is a sudden change in the transmembrane potential that travels along the length of the axon. As the action potential reaches the neuron's synaptic terminal, permeability changes in the membrane and triggers the exocytosis of ACH into the synaptic cleft. Exocytosis occurs as vesicles fuse with the neuron's plasma membrane. The ACH molecules diffuse across the synaptic cleft and bind to ACH receptors on the surface of the motor end plate. ACH binding opens the ligand-gated ion channels which conduct sodium ions through the cell's plasma membrane, changing the membrane's permeability. Sodium ions rush into the sarcoplasm. The sudden inrush of sodium ions results in the generation of an action potential in the sarcolemma. Now we will more specifically talk about the excitation of a muscle fiber. When the action potential reaches the triad, it triggers the release of calcium from the cisternae of the sarcoplasmic reticulum. The change in the permeability of the SR to calcium is temporary, where the terminal cisternae overlap. The thick and thin filaments of the skeletal muscle fibers interact as the effect of calcium release is almost instantaneous with the sarcomere. Troponin keeps the active sites inaccessible, while calcium is a key to opening the active sites. Troponin binds to both actin and tropomyosin. The tropomyosin molecules cover the active sites and prevent interactions between thick and thin filaments. Each troponin cell also has a binding site for calcium. When this site is empty, the muscle fiber is at rest. Calcium bonding changes the shape of troponin and actin. As the troponin molecules change shape, the attached tropomyosin strand is rolled away from the active sites. And now it is time for the contraction cycle to begin. The contraction cycle begins with the arrival of calcium ions within the zone of overlap. Once active sites are exposed, the energized myosin heads bind to the active sites, forming cross bridges. The energy stored in the resting state is released as the myosin head pivots toward the M line. This action is called the power stroke. When the power stroke occurs, the bound ADP and phosphate group are released. When another ATP binds to the myosin head, the actin molecules are broken on the active site. The active site can now form another cross bridge. Myosin reactivation occurs when the free myosin head splits ATP into ADP and phosphate. The energy released is to recock the myosin head, which begins the contraction of a muscle fiber again. Now that I have just walked you through the steps of excitation-contraction coupling in a muscle fiber, I will summarize the events in simpler terms. Arising from the brain or spinal cord of a motor neuron, which carries the action potential to muscle fibers throughout a muscle, the action potential travels along the entire sarcolemma through T-tubules which hold dihydropyridine receptors. These receptors activate the ryanodine receptors which release calcium into the sarcomere. The calcium influx triggers muscle fibers to contract and slide together. Thus, we have the excitation-contraction coupling events. Thank you for watching my video on excitation and contraction coupling.